processes that drive either acidification of vesicles, as we talked about already, or the production of ATP, which happens on the mitochondria, both being done by pumps of this sort. Okay? All right. So we'll start today. We'll talk about how cells communicate. I want you to remember again that cells are just collections of molecules. So when we say cells communicate, we're saying one collection of molecules that are bounded by membranes communicating with other collections of molecules or the environment and so on. But this collective set of molecules inside a membrane, which we call the cell, does remarkable things that look like it's moving, look like it's secreting, and so on, which we define as life, essentially. So again, the big picture, this should be fairly simple, and it parallels your lab manual for Daphnia. Cells communicate with each other via receptors, and these receptors bind signaling molecules called ligands. And this is a broad statement which actually has many, many, many different kinds in them. There are many different kinds of receptors, many different kinds of ligands that we'll talk about. So what that ligands bind receptors? After that, you can have two different kinds of outcomes, either a rapid change in protein function or a slower change with like the changes in gene expression, where new proteins are getting made inside the cell just in response to some ligand binding to some receptor. And when we think about ligands, we can think of them in terms of whether they are soluble in water or not. So if they're soluble in water, they're hydrophilic, they like water. If they're not, then they're lipophilic or hydrophobic ligands. The hydrophobic ligands or lipophilic ligands can simply cross the membrane and activate. Remember, the cell is bounded by a membrane. They cross the membrane and activate intracellular receptors. But hydrophilic ones cannot cross unless they have transporters. And they bind surface receptors or transmembrane receptors that are located on the surface and then lead to signaling downstream. That signal, if a molecule binds to another receptor, needs to be changed in form from one form to another. This is the molecular interaction that then gets changed into some other form. That's called signal transduction, where one form of signal is getting changed into another, and it usually occurs through the activation or generation of what we call as second messengers, which then go on and activate effectors. This system really exists to amplify the effect. You can have one signal binding to one receptor, activating many second messengers, which can go on and act on many effectors. Okay? And in the end, we'll talk about three basic modes of signaling. Signaling through ion channels, which we introduced in the last lecture already. Enzyme-coupled receptors, which are enzymes that you've also heard about already, but they're directly linked to receptors. So when the receptor binds a ligand, the enzyme gets activated, and then it does substrate to product conversion and things like that. And we'll finish with a class of receptors called G-protein coupled receptors, which are probably the best studied in biology. Okay? And also, as a result, or the other way around, if you will, they are being targeted by many, many drugs to alleviate many disease states. I'll talk about that in the end. So this gives you the broad idea of what we're talking about. That doesn't mean that everything that is to be discovered about signaling has already been discovered. This will be a theme throughout my lectures, as you will see, that all the things that I'm going to lecture about are our current summary of what's going on now. But that doesn't mean this is the only thing that's going on. In last lecture, I said in my lab we study double-stranded RNA going from one cell to another. Because the RNA has a specific sequence, that's also a form of signaling. One cell is signaling to the other which sequence to inactivate and so on. So that's at the infancy stage. It'll take us many more years before we figure out how that works. So I want you to have that idea that there are still many more things to be discovered, and what we'll talk about today is the summary of the process of signal transduction. This should be really familiar from your lab manual that cell communication can be thought of in three broad steps. First is the reception of whatever signal is being sent by a cell. And then the transduction. This is the process where the information is getting converted from one to another. And then finally the response of the cell. Whatever happens to the cell by the transduction pathway activating some effector. Okay? And when we think about all these components, we can think of them in many different ways for each of the steps. For the signal, for example, we can think about what kind of a signal is it. Is it hydrophobic? Is it hydrophilic? How did it get there? Was it just secreted out, or is that itself attached to some other cell, and so on? And what's the consequence of that signal binding to the receptor? When we think about the receptor, we can think about, is that on the membrane, like it's shown here, or is the receptor somewhere inside the cell? Okay? The general scheme still holds when the receptor is inside the cell. And what kinds of receptors are they, and how do they respond? The transduction pathway is essentially a relay mechanism. You can think of it as the signal is some sort of baton that's being passed on, and then other messengers carry the same baton and pass it on, or convert it into something else and pass it on. So those relay mechanisms and how those molecules that do the relay mechanisms behave, that's something you can think about. And finally, the response of the cell. You can see from this diagram that it's several steps removed from the signal. Right? So the signal doesn't know what the cell is going to respond. And the cell's response may not know what this is. Right? So the way that cells have evolved, there is a link between the molecule that's on the outside and what the cell does. And if what the cell does allows the cell to survive, or the organism to survive, it keeps doing the thing again and again. So you can have completely different responses to the same thing on the outside based on what the cell is. Okay? So we'll talk about each of these steps in detail as we go through today. So first step, what about signaling molecules reaching their targets? The target simply being the cell on which the molecule impinges. Typically, extracellular signal molecules act at a distance, and we can think of how they get to that distance as a classifying mechanism. Okay? They are secreted typically, and we can think of three different mechanisms. The first of which, a question like this may be asked in your exam, what are the three types of signaling mechanisms? So the first of which is called paracrine signaling. You can think of that as just signaling that happens nearby. So a molecule is secreted, and it simply is bound by receptors on cells that are nearby. So it's a local mediator that then acts locally. Second one is endocrine signaling, which is signaling that happens at a greater distance. This is how hormones work. They're produced, say, by your adrenal gland or your pituitary gland. They get secreted into the bloodstream, and they can act on very distant organs. Okay, so when you have adrenaline, for example, your heart rate can go up, but the adrenaline is being made by the adrenal glands, which are located above your kidneys. So the long distance that the hormones can travel is signified by endocrine signaling. And the final form is synaptic signaling, which is what happens in our brain, and also how our muscles respond to signal from neurons. And here you can think of the signal or the neurotransmitter as having traveled long distances through the axon, because the neurons themselves are pretty long. We have neurons that go from the back of our bodies all the way to the tips of our toes. So that's a very long distance that these molecules can travel. Or the response of one cell can travel, and then you can have localized secretion of the neurotransmitter. Okay? So those are three different forms of signaling from the point of view of the signal, whatever the ligand is that's being released. You can also think of another form of signaling, which isn't really about going from one cell to another. It is the cell communicating with itself. it seems a little odd at first. Why would you release something and then respond to it yourself? This form of signaling is called autocrine, auto for the cell. It can happen where a particular cell releases a signal and then, based on the levels of the signal outside, begins to respond again to that. Okay? The context in which this is really important is development. Remember that process I talked about at the very first slide last lecture. You start with one cell embryo, and then you go on and give rise to all the different tissues. At some point, that one cell divides into a cell that then gives rise to, let's say, all of your skin. Okay? So that cell then, every time it divides, has to keep doing the same thing. So usually that's like a feed-forward mechanism where you keep doing the same thing. So that, under such situations, you can have a secretion of a signaling molecule that simply reinforces whatever the cell was doing already to make sure that it doesn't waver from that fate decision. So this is a unique case where a cell is sending a signal just to respond to it itself. Okay? That's autocrine signaling. And another way in which this signaling can happen really locally without being secreted. Right? We talked about 3 different kinds of action when the signal is secreted, but what if the signal isn't secreted? That would be when the signal is located on the surface of one cell and it's in intimate contact with another cell. And that's why it's called contact-dependent signaling. And I'll also give an example from immunology and an example from development or embryonic development. The example from embryonic development is notch delta signaling. This is literally what happens during development when 2 cells are duking it out with each other. One of the cells that has high levels of notch or high levels of delta would go on and do something different from the cell that has the other level. So they'll start expressing notch and delta, and whichever wins at having more notch or more delta will fix that fate. Okay? So that's an intimate competition decision that's being made by 2 cells that are next to each other, and that's done in this form where one of the signals is a ligand and the other is a receptor, but it then leads to alternative fates in these 2 cells. A second class where this happens is the T helper cell and B cell interaction, where let's say the T cell has determined what is the antigen to which antibodies need to be made, then it literally, you think of it as programs the B cell to, or selects the B cell for further production of antibodies. You don't need to know the details of what these processes are and how they work, but I just wanted to give you an example so that you can anchor the concept of contact-dependent signaling for your further reading. Okay, so once these signals bind, we can really think of many different things that the signals can do. The field of signaling is incredibly broad, and if you really think about it, at any given moment, in any given cell, simultaneously so many things are going on. So everything that I'm about to present to you is just an organized way of us thinking about what's going on. In a cell, simultaneously, way too many things are going on. Okay? So here's one way in which we can think about it, the overall action of what a signal does. So they can either act slowly or rapidly, and I think this is a figure that's slightly modified and present in your manual, so you should be really familiar with this one. You can think of the signal acting really quickly or really slowly. By quickly, we mean less than a second to minutes, and in fact, some new work on neurotransmitter release suggests that it can be on the order of milliseconds and less. Okay, very fast signaling, which is typically through the alteration of protein or through the alteration of something that is already there. You cannot make new things at that rate in cells. If you have to make new proteins and new cytoplasmic machinery, that has to be slow. It happens on the order of minutes to hours. Okay? So you can think of the speed with which a process happens It's either really fast or really slow. The other thing that you should remember about cells is the cells are not all located in the same environment. So in our body, the liver cells are in one place, the heart cells are in another place. Everything is in its place. And one of the ways in which that's ensured is that there are extracellular signals that can tell the cells simply to live or die. So a cell in isolation is not possible, basically. Okay? There has to be something on the outside, even if it is just isotonic solutions that you have in a petri dish, that's fine. But if there was a vacuum on the outside, the cells dead. So there are signals in the context of animals or plants which are local signals that help the cell to live. And if not, they have processes that cause programmed cell death or apoptosis. We'll talk about that later. So here's an illustration of that. You can imagine there's a cell, and there are, say, a complex of signals, ABC, which allow the cell to live. And if you don't have those signals, then the cell commits suicide or apoptosis, programmed cell death. And that's illustrated here. Now, the reason why this particular concept is important is because this then explains how different kinds of cells could be kept alive in different environments. So you can think of them as niches or niches, however you pronounce it, where there are signals on the outside that support just the ability of the cell to live. Okay? So signaling is fundamental. It's required for cells to live in our bodies. The second aspect that we can think, once we understand that even living requires signals, is after a cell is kept alive, now the cells can be induced to do different things. And for you to remember easily, you can think of it as 3 different Ds, either to divide, to differentiate, or to just do something. Okay? The divide part is where you want to proliferate and make more of the same. Our entire blood supply gets replenished at a rapid rate. You can look it up online what that rate is. It's remarkable. So that means cell division is an ongoing process that happens all the time in our bodies. So there have to be signals that instruct the cell to divide. Now, differentiate is a process where you go from one kind of cell to another kind of cell. The easiest context in which you can think about this is where stem cells, which can give rise to many different types of cells, are being induced to give rise to any one type of cell. So let's say there is a shortage of red blood cells, then you could have hematopoietic stem cells being differentiated to give you more red blood cells. In fact, that particular differentiation process is one of the most abundant differentiation processes that happen. And a third thing would be to just do something, like move or invade across a epithelium. Let's say there's an infection that has been detected and there are crawling immune cells that need to cross a membrane layer, not a membrane layer, a cell layer. That induction can happen through signals that are on the outside. So the way to think about it is you need the survival signals, the ABC from the previous slide, and you also need D and E that will help grow and divide, and F and G which might help differentiate. So there are still remarkable aspects of this that are being discovered. I think as of last week, there was this paper which was just really cool, so I'm sharing this with you. It was published in the journal Science. But there was one kind of cell called a glial cell, which is about 1/3 of the cells in your brain. And they showed that that particular cell can divide and now give you a neuron, which is a case where 2 things are happening simultaneously. You're getting division and differentiation at the same time. So glial cell dividing to give you a neuron directly. Okay? So it's the tip of the iceberg. We don't know how that happens. We'll figure out as time goes. But this was as of last week. So all these, so in terms of do, it can be any kind of thing that it does, specialized functions like secretion, movement, anything that you can imagine. Everything is induced by a signal on the outside. Okay? So far what I've said should give you an idea of how we're building up hierarchically. So within a cell there are just bunches of molecules, but because they're surrounded by a membrane, it makes them one organized unit, right? And then you can move that unit about by having signals on the outside. That signal from the outside moves the entire unit, the whole cell, to one place to do something or to change it to another thing, and so on. Okay? All right, the third thing that we can talk about is now from the cell's point of view. So far it was from the signal's point of view. Now you can think of the cell's point of view. Just because some random signal is coming from the outside doesn't mean that the cell will every single time do the same thing. Not true. The cell interprets that signal. Okay? That means for one ligand binding one receptor, the outcome of what the cell does depends on what type of cell it is. We'll illustrate it using, I think, acetylcholine receptor, where the receptor protein can still be the same, but based on which cell type the receptor protein is located on, the cell does something completely different. So here's the example on the right. So on the right, there's the molecule acetylcholine. The receptors are found on muscle cells, smooth muscles, on skeletal muscle cells, and also on the salivary gland. Okay? So clearly there are very different types of cells that have specialized molecules and things in them, but the response of activation is very different. So if it binds the heart cell, you have a decrease in heart rate. I think this was also probably one of the drugs you added in your lab. The skeletal muscle, if it binds to the skeletal muscle receptor, you get an opening of an ion channel and contraction of the muscle. In the salivary gland, you get secretion. So what happened is entirely dictated by what the cell is, right? And the collection of molecules that were present in the cell. So if I ask the question, what does acetylcholine do? Your correct answer should be, it depends. You cannot say it does this or it does that, because it depends on what the cell is that it's acting on. Okay, so just to summarize again, the receptor proteins can be unique on each cell. The intracellular machinery with which the cell interprets, that can be different. And based on that, you can get the same signal having completely different outcomes in different cell types. Okay. All right, so the fourth type of overall action that we can think about is how the signal itself isn't one thing. The signal has many components. You can think of it as it has charge, it has shape, it has it has hydrophobicity, hydrophilicity, etc. But that's all for each single molecule. And you can imagine, based on charge, there will be a membrane effect, membrane potential effect. Based on the shape, there could be something that binds. But another important concept to remember is based on concentration, there can be something completely different that happens on a cell. So even having a receptor on a cell that binds a certain ligand doesn't allow you to predict completely what the cell does, because it will depend on the affinity of that interaction. So something that binds really, really tightly to a ligand will bind even when you have very small amounts of the ligand. We talked about KD in previous lectures. So you can imagine something that binds a ligand really tightly will be activated very quickly. But something that binds a ligand very weakly will require a lot of ligand to get activated. So that means, based on the concentration, you can get different effects. Okay? So here's another example from embryonic development. In embryonic development, there's a concept called morphogen. Morpho as in shape, gen as in making, or as genesis, and so on. So morphogens are molecules that people think of as being in concentration gradient across a flat surface. So let's say you have a sheet of cells, and you have to have the first cell in that sheet do one thing, and the last cell do another thing. You could achieve it by having different ligands all along, or you can have one ligand in different concentrations all along. And you have the receptors responding differently all along that surface to the concentration of the molecule. And so you get different fates based on the concentration, and here's an illustration of that. If, let's say, there's a source cell that's just simply dumping out to the extracellular space a molecule, which we call the morphogen, then it'll be very high concentration near the source, and because of simple diffusion, it'll be weaker concentration further out. So the cells that are located, which are all of the same type, all along that gradient will experience the molecule differently. These guys experience very high concentration. These guys experience very low concentration. And that can be read out by the organism to give you different responses all along the length of that morphogen gradient. Okay? So that's it on the different ways of thinking about just the signal and the cell, in how the cell and signal respond during cell communication. But there are specialized cells which can receive many different stimuli, which we kind of hinted upon when we were talking about how receptors could be activated. There was mechanical activation, thermal activation, heat activation. Well, all those different kinds of receptor activation that we talked about in last lecture could be used by cells to respond to all sorts of different things. How could a cell respond to heat or a cell respond to light? The way it does that is by having receptors that respond to heat and light, molecules that respond to heat and light, and then transducing that signal to then do something. So you can have, at the level of cells, behavior, where if you had a cell on a petri dish and then you put some molecule at one corner, the cell will crawl to the molecule. That's because the receptor binds the molecule, activates downstream signaling, which leads the cell to crawl. Okay? So one of the things that is used when crawling is adhesion to the substrate. It needs to stick to the substrate. We already talked about membrane distortion in last lecture for how mechanical forces are transduced. And sound itself is a mechanical distortion for at the level of the cell. Okay? If you have sound that's produced with sound waves, it can distort the cell quite a bit. So that's many different forces, but the most widely used and the best understood, simply because it's the easiest to study, are chemical signals. And chemical signals can be of many different kinds. You can imagine hydrophobic, hydrophilic, all the things that we talked about, including dissolved gases and steroid hormones. So here's where what I said at the beginning, that receptors don't have to be on the surface, is important. So even though oxygen, nitrogen can just diffuse across the membrane, cells have receptors with which they can respond to these molecules that they find inside the cell. Okay? And steroids, which can cross the cell membrane, cells can respond to that too. And you'll also notice there are things like amino acids, which are building blocks of proteins. They are doing double duty as these signaling molecules. Like glutamate is often used as a neurotransmitter. Okay? And adenine can be found outside, nucleotide residues can be found outside and act as signals. Okay? So any small chemical that can bind to a specific receptor can act as a signal. So we'll talk about lipid-soluble ligands to begin with, and some of the most famous ones are the steroid hormones. And the chemical structures of these ones you don't have to know, but it would be good for you to keep in mind that the steroid hormones all come from cholesterol. They're made from cholesterol. Here's an example: cortisol, estradiol, and testosterone. Cortisol is the famous stress hormone. Probably have seen many ads and things on TV about how cortisol is bad for you. But it's the stress hormone, which also is derived from cholesterol. Estradiol and the testosterone are the sex hormones, one's in the female and the other's in the male. but both are actually found in male and female. It's just one dominates in the female, the other dominates. And you also have other hormones that don't look like steroid hormones. So only steroid hormones are made from cholesterol, and they can cross lipid bilayers. But other famous hormones include retinoic acid, thyroxine, which is made by the thyroid gland, and vitamin D, which is made the skin in response to UV. So here's a summary of what I told you so far. Their structure can vary. Steroid hormones are synthesized from cholesterol. Cortisol, which is made by the adrenal gland, influences metabolism, which is a stress response. Sex hormones that are made in ovaries and testes, which is estradiol and testosterone. They determine the secondary sex characteristics that distinguish male from female. And vitamin D, which is made in the skin in response to sunlight, regulates calcium. And we'll come back towards the end of the lecture about calcium and many things that calcium does. And it regulates uptake and secretion. And thyroxine, which again, remember, is a non-steroidal hormone, and it regulates metabolism as well. Okay. All right, so that's lipid-soluble signals. Because the lipid-soluble signals can enter the membrane, the receptors are often found inside cells. So they're found in either the cytoplasm or they're found in the nucleus. In either case, when the lipid-soluble molecule enters the cell and binds the receptor, there's usually a translocation step that goes into causes the protein to go into the nucleus, and then you have activation of sets of genes that happens in response to it. Now you can imagine, because it's lipid-soluble, it will not be able to circulate freely, because our circulation is mostly water. So the ligand itself is often carried with the help of carrier proteins, which can bind to hydrophobic things and carry them through fluid. So here's an illustration of how a steroid hormone receptor might work. They're usually transcription factors. We'll talk some more about transcription factors in later lectures. For now, you can think of them as just proteins that bind to DNA and cause the production of mRNA, okay? Or gene regulatory proteins. That's the second term that is used in your textbook, as an alternative for transcription factor. So typically, they're held inactive by binding to some inhibitory protein. Then, when the steroid hormone binds, let's say in that pocket as illustrated here, that changes the conformation of the protein such that now the inhibitor can no longer bind. And it also induces the translocation into the nucleus of this particular protein. And in the nucleus, through its DNA binding domain, it could bind to a receptor binding element. And when that happens, you can have transcription of target genes or the production of mRNA for target genes downstream. Okay. So to summarize what I just told you and to revise it in your own head, nuclear receptor subfamily is one of the most common steroid receptor subfamilies. They're bound to an inhibitory protein and are held inactive. The binding of the ligand causes a conformational change, and that results to the translocation usually into the nucleus and then binding to the DNA. And transcription is then increased, which then leads to specific responses within the cell. Okay. So one thing very important that you should remember is these receptors are intracellular typically. They're not on the surface. And as you can imagine, there is, in this case also, many different responses that are possible, even though the hormone is released into the circulation. And this is particularly important because you can imagine that a signal like an endocrine signal, which is simply released into circulation and therefore has access to every single type of cell in your body, you can't have all the cells do the same thing at the same time. then the homeostasis of the body will not work. So the only way that that's determined is by having different cells having different receptors that are differently able to respond to the same hormone. Okay? So that's what dictates what the response is to a hormone that's secreted. Okay, now we'll move on to lipid insoluble signals. And as I mentioned, these by definition have to be outside the cell, can't cross the cell membrane unless there is a particular transporter for it. And this is the most commonly studied and sort of extremely well understood class of signaling, where you have small polar water-soluble molecules, so they can simply be in circulation. They don't need a carrier protein for keeping them in solution. And most neurotransmitters are like that, peptide hormones are like that, growth factors that keep a particular cell growing in a particular environment, they are like that. Everything we smell can be thought of like that. Tastants, everything we taste usually does not cross the membrane in our tongues. And all of them then would have to bind to cell surface receptors and then lead to downstream signaling. Okay? So that's schematized here. There's something on the surface binds to the receptor, and then you have downstream signaling. So before we go on and talk about the three different kinds of signaling and downstream signaling, any questions on hydrophobic ligands, hydrophilic ligands, and general mechanism of how cells respond to signaling? Was it too fast so far, or was it okay? Too fast? Too fast still? Okay. So I'll try to slow down some more. Now what we're going to talk about are three different types of signaling, and then we'll spend some time in the end talking about the details of signaling that occur. Okay? So in terms of the pace and what I can do to help with covering some of the material that we need to cover, I'll make sure that I put the lectures for the next week, the Sunday before, and if you peruse it before the lecture, it might make it easier to follow. One of the problems with cell biology is the subject itself is incredibly vast. So we can do one of two things. We can ignore half of it, which we already have, by the way. There are many things that we have already ignored. Or we could try to cover it in a sort of abbreviated fashion. So we try and give you a broad range of things which will form a basis for many other things you'll learn going forward. So I would advise, do try to peruse the lecture material before, and I'll stick very close to the PowerPoint so that everything that you are reading already will help you to reinforce what you're learning. The last one should be a 3. But anyway, so the ligand-gated ion channels, or ionotropic receptors, enzyme-coupled receptors, and G-protein-coupled receptors. So just to get a broad view, this is simply 3 different kinds of things that you find on cell surfaces, and their name already suggests what they're going to do. So we'll quickly go through what that is. Ion-coupled receptors, we've already talked about. You have ions that are trying to come through down their concentration gradient. Remember, they have to move down their concentration gradient if they're going through channels. And the channel is closed. And for the channel to open, you need the gating process where a small signal binds and then gates it open. And when that opening happens, the ions can rush through. Okay? So that's ion channel coupled receptor. So you might think, what's the difference between ion channel and ion channel coupled receptor? It's just that the ion channel coupled receptor binds something and then opens. It's an ion channel that's always just a hole, doesn't have to bind anything. It can just be a hole. So it's typically involved in synaptic signaling between neurons and other excitable cells, are open and closed by a small number of neurotransmitters, alter ion permeability and membrane potential changes because it's in neurons. The second type is enzyme-coupled receptor, and this again, as the name suggests, there's an enzyme that's stuck to a receptor. So there's a transmembrane domain protein which has an inactive catalytic domain, And how would you couple ligand binding to activation? It's often through bringing these two inactive things together. And what happens typically is this domain will phosphorylate that one, and this one will phosphorylate that. It's called cross-phosphorylation. We'll talk about a particular example of that shortly. And that then leads to activation downstream. So here's a case where the enzyme is itself stuck to the same protein that is passing across the membrane. Here's another example where the enzyme itself is not found in this dimerizing partner, but then once dimerized, it binds with another protein which is associated, and that is the enzyme. So there are two different ways in which you can think of an enzyme being coupled to a receptor by binding, the enzyme being coupled by directly being part of the same peptide. So single pass transmembrane proteins are typically used in this form, and they have many different kinds of structure. Either function as direct protein kinases, which is what was shown here, or associate directly with and activate other proteins, which is what's shown. So the point of having something right up there that's an enzyme, right at the reception of the signal, is it gives you an advantage to do many different things in response to one. So enzymes are typically multiple turnover, right? They convert a substrate to a product, they convert another substrate to another substrate, and they can keep doing that. So to one binding event and one activation, you get a massive amplification. And that is often further increased by having many more things attached together. So you can get a cascade effect. Okay, and the third one, which is really, you could think of it as also a specialized form of enzyme coupled receptor, but this is so vast and so important in biology that it gets its own name, G protein coupled receptor. Because what it's coupled to is a class of proteins called G proteins because they bind GTP. And this is unlike what you've already heard, like Rho, RAS, and RAC, and all these small GTPase proteins. These are called heterotrimeric G proteins. We'll talk a little bit more about them towards the end of today's lecture, but they, as you can see, look different. They have three different things that are stuck together, hence the term heterotrimeric, okay? And they have an inactive receptor, let's say, and the receptors are typically 7 transmembrane domain receptors. They have 7 transmembrane domains. And then you have an inactive enzyme. So this inactive G protein is an intermediary between the receptor and the enzyme. Okay? Usually in the previous case we saw there's just the receptor and the enzyme. There's nothing in between. Here you have the inactive G protein that's an intermediary. What happens is when the signal binds, the G protein coupled receptor complex is formed and the G protein is activated, which then goes on to activate other enzymes which will act further downstream. We'll talk about this in greater detail towards the end of today's lecture. So it's a very large family of multi-subunit transmembrane proteins. So in the organism that I study, there are thousands of them. And that's in an organism that only has 20,000 genes. There's way more than 1,000 G protein coupled receptors alone. So you can see how many different such things they use. They indirectly regulate the activity of the nearby target protein. That's why I said there's an intermediate. Okay? And they contain the GTP binding protein complex. So that's the middleman that carries the information from the receptor to the enzyme. And these are G protein coupled receptors. Okay, so once you have activation at the membrane through these 3 ways, say ion channel receptor or G protein coupled receptor or enzyme coupled receptor, you can think of those cell surface receptors as first messengers that carry the signal. And then you have second messengers, and then you have effectors. There are some rare cases in which there are even tertiary and quaternary messengers, but we won't talk about those. So a combination of things can happen after that, which leads to an amplified signal, many, many different things. And that's second messengers are usually far more abundant than the first messenger in number, just number. And therefore, they can then activate many different effector proteins. So that's illustrated here. You have an extracellular signal that binds a receptor protein, and you have a set of intracellular signaling proteins. We'll talk about those next. And that can lead to a final set of effector proteins, which can cause changes in metabolism, like the cortisol case or the thyroxine case that we already talked about, or changes in the gene regulatory protein, like the one example we talked about is when a steroid hormone binds its receptor, so there can be other transcription factors that are activated, or changes in the cytoskeletal protein, and that you can imagine would be required for a cell to crawl, right? If it has to crawl in a certain direction, it has to change its cytoskeleton. So those sorts of changes can happen in response to ligand binding on the extracellular surface. Another concept that's really important when you have a relay mechanism like this are switches. So you can have molecules that are kept off in terms of signaling and then turned on in response to some relay. These should be quite familiar to you now because these are the changes that can happen to protein, which we've already discussed in this course. Like phosphorylation by a protein kinase, that could act as an on switch, which turns on a protein to then do other things. Dephosphorylation of that same protein by a phosphatase, which turns it back off. Okay. Another way in which you can have a switch being thrown in response to a ligand that binds extracellularly is switching the small molecule that is bound to another protein. So in the case of small GTPases, like Rho, RAC, and so on, they are off typically when they are bound to GDP. When the signal comes in, GDP is exchanged for GTP, which activates that protein. And let's say Rho does some cytoskeletal rearrangements, and after that, when it hydrolyzes GTP, either on its own or helped by something else, the signal stops and you go back to the off position. Okay. So things like kinases and phosphatases, things like guanine nucleotide exchange factors and release factors, they help throw the switch, go from on to off. Now, in addition to just having the signal come, bind to a receptor, and then respond to it, in different cells, the response molecules could be arranged in such a way that the response is incredibly fast. The cells have ways of arranging molecules to minimize just diffusion being the cause of something. Because diffusion is this random process, and in some cases, when you have large molecules, it can be pretty slow. So sometimes you have proteins arranged together as signaling complexes, and the goal is to make the whole process faster, make it more efficient, and to also achieve specificity in the signaling response. And for that, when I'm showing you all these different pictures, it could look like there are many different things that are going on, and you have to pay attention to this and the other, and the shape of the particular molecule and so on. But don't worry about all those things. The most important message that you should be getting across from each picture and each slide that I'm talking about is what is the concept that's being talked about and how is it achieved. Okay? So the concept that we're talking about here is how the cells are responding quickly to a signal, and the way it's achieved is by just having a platform or a scaffold protein on which you stick all the ones you want to respond. Okay? So you can imagine if you have a series of inactive signals, there are 1, 2, 3 inactive proteins that are held together. You have the signal molecule bind the receptor, and then simply that signal is passed on. It doesn't matter what the identity of these particular proteins are. You can think of this as, say, collecting together a few kinases, and then all at once you activate all 3 of them. Then they can go on and activate their substrates. Otherwise, you wouldn't have been able to activate all 3 to the same extent every time in response to this molecule. Right? So this gives you something like a combinatorial control. by having the collection of kinases that you want to activate together. A second thing that can happen is that this particular scaffold gets formed only when the ligand binds. So let's say before the ligand binds, there are no inactive proteins that collect together, but the binding of the ligand changes the conformation or changes phosphorylation status, and that now recruits a whole bunch of these other guys. Okay? And then they all get together activated and do downstream signaling. So in both these slides, the concept is very simple. You just collect the proteins together onto a scaffold, activate them so that the activation happens much faster than if you're waiting for one by one to activate. Okay, so that's the concept of having complexes, signaling complexes, or scaffolds together. The scaffolds don't have to always assemble only on proteins. Remember that the cell membrane itself has all sorts of molecules there that give it subdomain type structure. And a very famous and well-studied example is the assembly on phosphoinositides. So these are PIP2, which you would have heard of already in Dr. Fraulitz's lectures. I'll talk about it again in the next slide, I think. You have, let's say, an inactive receptor. Let's say a ligand binds to it and leads to the phosphorylation of PIP2, let's say, and you make PIP3. Then that PIP3, which is present locally, imagine the cell surface, it just bound that receptor, it just phosphorylated all the PIP2 around it. So now there's a patch of PIP3. That recruits these other downstream signaling proteins and leads to activation. So these would be three different ways in which you're concentrating downstream proteins for activation in response to the signal. So now we'll talk about some famous cases of signal activation. One of the most famous ones are receptor tyrosine kinases. And in fact, one of the earliest cancer genes that were discovered was a mutation in this, which made it overactive. So if you have too much signaling, you can imagine if the signaling is coupled to divide, a cancerous cell could just keep dividing. So these classes of proteins have growth factor effects often. You don't have to know all these acronyms, but if you look across, you see many of them are GFs. So EGF, IGF, NGF, and so on. The GF is growth factor. And the one that says SF is actually stimulating factor, macrophage cell stimulating factor. It's called the stimulating factor. So that is also a cell division molecule. So these proteins are single-pass transmembrane domain proteins. You don't have to know all these domain structures, but you just get the general idea of what they do. They bind some ligand, and they have a tyrosine kinase domain intracellular. And when the ligand binds, the tyrosine kinase domain is activated, and the tyrosine phosphorylation can happen. So already we've come across how this could happen. It's through dimerization, typically. And when you have dimerization happening, you have cross-phosphorylation. That is, this one will phosphorylate that one, and that one will phosphorylate this one. So that cross-phosphorylation activates the receptor. Okay? Receptor tyrosine kinase is activated. Once that's activated, you can imagine recruitment of downstream signals, as we already talked about, which could themselves be enzymes, or which could simply be proteins that bind to other proteins. An example is SH2 domain, where SH is Src homology. Src happens to be a kinase. You don't need to know that. You just need to know that SH2 domain is an example that can bind to the phosphotyrosine. So it won't bind to the receptor unless the tyrosine is phosphorylated. But when phosphorylated, then it can bind and lead to activation further. So one of such things, such a protein that can be activated in response to receptor tyrosine signaling is called phospholipase C, beta. From the name, you can guess what it's going to do. It's a lipase, so it's going to cleave off lipid things. Okay? Phospholipase, so it's going to cleave off phosphorylated lipid things. So here's PIP2, which has a diacylglycerol and the inositol triphosphate attached together. It cleaves that bond, releasing diacylglycerol and inositol phosphate. So think about that. That was one enzyme that cleaved one substrate and now has two products, one of which is hydrophobic and therefore will be restricted only to the membrane. The other, which is soluble, will be in the cytosol. Okay? So this is a case where both those things can go on, both second messengers can go on and activate their own substrate. So we'll talk about IP3 first. IP3 binds to a channel on the endoplasmic reticulum and releases intracellular calcium stores. So you remember from last lecture that in the cell works really hard to keep the cytosolic calcium levels very low, 0.1 micromolar, right? But the endoplasmic reticulum and the extracellular calcium levels are very high. And here's how it's able to use that store. In response to a signal, there is a rush of calcium that comes out, and that can activate downstream processes. So you can imagine this as some sort of check. This particular protein is activated only when you have diacylglycerol and you have intracellular calcium. So you have two different activators that are used to make sure that it's a bona fide signal before you have further phosphorylation. Okay. Now, here's a closer look at what I just told you. Here's the structure. You don't need to know the structure. You just need to know that there are two fatty acid chains that are in there, and once TIP2 is cleaved, you get diacylglycerol and IP3. This stays in the membrane, plane of the membrane, and this goes into the cytoplasm to activate the release. Okay. Now, even through receptor tyrosine kinase, one of the examples that I gave you so far was where the scaffold was on the membrane, and it recruited many other things on the membrane. But you can have enzymes that are then activated in response to receptor tyrosine kinase signals. So a famous one is RAS, which you should have already come across, but perhaps not in this form. So RAS is another famous cancer-causing signal which, when hyperactive, leads to cancer. It's found tethered to the membrane. So when a receptor tyrosine kinase is activated, you don't need to know the names of any of these scaffolds that are here, so don't worry about it. But you know what they are. They're basically SH2 domain containing things that then bind only to activated receptors, right? And then you have a bunch of other proteins that interact with, one of which is a guanine nucleotide exchange factor, or GEF, which changes the GDP for GTP. Remember, we talked about how a signal comes in and then it can turn something on or off by just switching the small molecule that is bound to another protein. So here's RAS bound to GDP. You switch GDP off, put in GTP, and now you have an active RAS, and that leads to downstream signaling. Okay? And this can happen in the plane only for the RAS that is nearby. So you can imagine domains of cells that are being activated, not the whole cell being activated. And when this RAS is activated, it can then go on to activate more phosphorylation. So this is another famous cascade effect of phosphorylation. As I told you, that signal relay can lead to a cascade effect. It's called mitogen-activated protein kinase. Mitogen, mitosis promoting, so it causes division. So you can understand how this being inappropriately active would lead to cancer because you have too much cell division. Okay? And that happens because the active RAS activates this one, which then activates another MAP kinase, which then activates the MAP kinase, and finally you get this activation. So you have MAP kinase, activating MAP kinase, which then activates MAP kinase, which then activates proteins that regulate gene expression. Now, there could be other MAP kinases out there, but this is the best study that we know of where there is this relay of 3 different kinases, where there's a series of phosphorylation that leads to a really amplified effect from the membrane all the way into the nucleus. So in the last few minutes, we'll finish with thinking about G protein coupled receptors in greater detail. So far we talked about enzyme coupled receptors, very briefly about ion channels, and they did feature in the response to tyrosine kinases. Now we'll talk about G protein coupled receptors. As I mentioned, there are always 7 transmembrane domain proteins. They even typically do have this large intracellular loop, but you don't need to know that. And if you look at all the drugs that are out there in the market for any disease, if you took all the drugs that are out there in the market and asked what do they target, and you make a pie chart of what kind of proteins do they target, number 1 there, with 27% of all drugs, anything that you can think of, are rhodopsin-like G protein coupled receptors. Rhodopsin is the G protein coupled receptor in our eye that allows us to see. So as I said, it's a very broad class of molecules. That's why they get special attention. Up next are nuclear receptors. Okay? You can imagine why. They are intracellular already, and then you have ligand-gated ion channels, then voltage-gated ion channels. So everything you heard in today's lecture is 50% of all the drugs that act on the human body to help with any disease. Okay? 50% covers all of this. The other proteins are also very interesting, but we're not going to talk about them now. So G protein coupled receptors, at the heart of it is the heterotrimer, and like the RAS G protein or any other small G protein, the heterotrimer also has a GTPase cycle. There's an activation-inactivation cycle that happens. So in the inactive form, it's bound to GDP. It's an alpha subunit that's bound to a GDP, and these two are called the beta-gamma subunits. These squiggly bits that you see up there are lipid modifications that allow this otherwise soluble protein to be tethered to the inner leaflet of the membrane. And when you have an extracellular signaling molecule, it helps kick off the GDP for GTP. But you can imagine that's happening not because the extracellular signaling molecule entered the cell, but through conformational changes that are transported across, or not transported really, communicated across the structure of the GPCR itself. Okay? So when it's activated, you have GTP bound alpha subunit being separated from the beta gamma subunit. Now you have, again, 2 subunits that are produced, the alpha that's bound to GTP and the beta gamma. And in general, both these can go on and activate other downstream signaling pathways. It's much like diacylglycerol and IP3. You generate 2 upon activation, and both can go on and act on downstream activators. So here's that example for adenylate cyclase as the enzyme that we briefly mentioned before. You have the alpha subunit. In this case, it's called alpha S for stimulatory. It stimulates the adenylate cyclase. And when the alpha S subunit activates the adenylate cyclase, it leads to a cyclization reaction, which leads to the production of cyclic AMP. And the cyclic AMP is a second messenger, meaning it's in many, many, many copies, and it's a small molecule. Okay? This second messenger then greatly amplifies what's happening at the membrane, and it could be bound by a protein, like an inactive protein kinase, as shown here. This should be very similar to what we already saw, where there was a protein kinase C that was binding to diacylglycerol and to ions, calcium ions. Here you have protein kinase A, or pKa, A for cyclic AMP, that binds, its inactive domain binds it, releasing the activated protein kinase A, and the protein kinase A then goes into the nucleus. Okay? We'll talk about that in a moment. Here's the reaction for how cyclization happens. And once this cyclic AMP is made, there's another enzyme called phosphodiesterase, which can release the cyclization to give you 5' AMP, and that signals the end of the signal. Okay? So the activated protein kinase A enters the nucleus, and within the nucleus, it can activate other proteins. You do not need to know which proteins are being activated and what's happening, but the broad picture that I want you to remember from this is you have a G protein coupled receptor. It has heterotrimeric G proteins. How the heterotrimeric G protein is activated, you should know. And then you have activation of adinylate cyclase, which leads to the production of cyclic AMP, which then activates protein kinase A, which enters the nucleus to do stuff. Okay? You just need to understand how that signal cascade works. Okay, a second kind of G protein coupled receptor mechanism is illustrated here, where now the beta gamma and the alpha from another form called GQ. The previous one was GS, right? Activates another enzyme. This time the activated enzyme is phospholipase C, the one that we've already seen, right? So you can see that different signals can activate the same enzyme. We saw before that the receptor tyrosine kinase could activate phospholipase C. Now you have G protein coupled receptors also being able to activate phospholipase C, which leads to the same downstream effect that you saw already. So in this case, you should know which are the second messengers. The second messengers in this case are calcium and IP3. In the previous case, the second messengers would have been cyclic AMP, right? So make sure that you understand what the second messengers are, and you should be able to articulate why this is advantageous, because the diacylglycerol is restricted to the membrane and the IP3 is soluble, and therefore there can be signaling at both places. So to finish, you'll notice that in all these signaling mechanisms, there was that release of calcium that happened into the cytosol. And the cell uses the release of calcium for a diversity of processes. We won't have the chance to go into every single thing, but I hope that this list and what I'm going to tell you about it will give you a context for all the different processes that you've read so far and you will be reading in the future. Like muscle contraction relies on calcium release. That released calcium is critical for cell division, because then there are other proteins that respond to cell division. Secretion, whenever vesicles are fusing to the plasma membrane and secreting, they typically require intracellular calcium because there's some protein there that binds to it. Endocytosis, when things are being taken up into the cell, calcium is needed. Fertilization, the very beginning of multicellular life, requires calcium. In fact, there are classic movies where when a sperm fuses to an egg, there's a wave of calcium that gets released. And you have synaptic transmission, which activates calcium in the downstream cell. All of metabolism, because there are many enzymes that respond to calcium levels in central metabolism. And cell movement, because calcium powers the cytoskeletal rearrangement through other proteins that bind to calcium. So I think that's it for today. Thank you for your attention.